Motor neurons are extraordinary cells of the central nervous system, which extend from the spinal cord throughout the body. They control muscle function and are required for movement and breathing. The survival of motor neuron protein, or SMN, is essential for the correct development and function of motor neurons, and is key to normal muscle function and growth. A reduced amount of SMN causes a devastating disorder that affects approximately 1 in 10,000 newborns, spinal muscular atrophy, or SMA. In healthy individuals, there are 2 nearly identical genes coding for SMN, SMN1 and SMN2. Most of the functional SMN protein is produced from the SMN1 gene. The SMN2 gene, on the other hand, has alterations in its DNA sequence that leads to a defect. This defect has consequences in a regulatory mechanism called splicing. As a result, this gene produces only a limited number of functional mRNA and only 10 to 30% of the functional protein. In individuals with spinal muscular atrophy, the SMN1 gene is inactivated by a mutation or deletion, so that it cannot generate SMN protein at all. The SMN2 gene does not produce sufficient SMN protein to maintain motor neuron function, resulting in muscle wasting and severe mobility impairment. In a collaboration between Roche pRED, PTC Therapeutics and the SMA Foundation, we discovered orally available molecules that may selectively correct the splicing defect of the SMN2 gene. This may allow sufficient functional SMN protein to be produced, even if the SMN1 gene is inactive, as is the case in spinal muscular atrophy. In animal models of SMA, the compounds allow the animals to survive, grow and develop a normal neuromuscular system. Standing, walking, breathing, things we take for granted are a struggle for individuals with spinal muscular atrophy. We work to make a difference for these patients and their families.